Thank you, Lori, for the introduction. My name is McFadden, as she mentioned. I'm a graduate student at Duke University in Amanda's Argonaute lab, and I will be introducing our speakers today. Our first speaker is Brianna Bibel from the Joshua Tor Lab at Cold Spring Harbor, and I invite Brianna to share her screen and begin her talk.
Hi, everyone. My name is Bibel, and I'd like to start by thanking the organizers for giving me this opportunity to tell you about my work studying the phosphorylation-mediated regulation of RNA interference, or RNAi. RNAi is an evolutionarily conserved mechanism of post-transcriptional gene regulation. It uses small RNAs to target specific messenger RNAs and decrease the production of their corresponding proteins. These small RNAs include a class of RNA called microRNA. These are typically about 22 nucleotides long, and they target messenger RNAs containing sequences typically at the 3' UTR that have at minimum complementarity to a 6 to 8 nucleotide long seed region in the 5' end of the microRNA, and some targets also have additional pairing in the 3' supplemental region. These small RNAs are loaded into a protein called Argonaute, or AGO, where they serve as guides, taking Argonaute to corresponding targets where upon binding, they're able to induce target repression through translational inhibition and mRNA degradation with the help of cofactors. RNAi is a crucial regulatory process, but there are still many unknowns about it, including how the protein at its center, Argonaute, itself is regulated. One potential way of regulation is through phosphorylation of a region of Argonaute termed the eukaryotic insertion, or EI. Within this flexible region is a cluster of potential phosphorylation sites. Golden et al. And Huberto et al. Both independently found that this cluster gets phosphorylated in a variety of tissue types and organisms and seems to affect Argonaute's ability to interact with target messenger RNAs. Golden et al. Furthermore found that this phosphorylation was carried out by a kinase called CK1 alpha and removed by the phosphatase PP6. If they disrupted this phosphorylation cycle, they saw a global dysregulation of microRNA-mediated repression. But the mechanism was unknown, so I decided to see if I could figure out the mechanism. You can imagine phosphorylation affecting Argonaute in many different ways. AGO has to first bind to the guide microRNA, then bind to the target, and then release that target before it can bind to another one. And you can have phosphorylation or dephosphorylation occurring at any of these steps. To break things down, I developed an in vitro kinase assay using recombinant human CK1 alpha and human AGO2. I introduced the kinase to Argonaute when it's either RNA-free, bound to just the guide microRNA, or bound to both the guide and the complementary target. For these experiments, I'm using a natural guide-target pair as a template for further investigations. When I carried out this experiment, I found that only in the presence of both the guide and the target was I able to see phosphorylation, implicating target binding in inducing phosphorylation. I wanted to see what it was about the target that was making the effect. So I decided to try to figure out the minimum requirements of the target for inducing phosphorylation. I started with target length. I shortened the target from the 5' ends. And when I did this, I found that I could still see phosphorylation at 14 nucleotides, but not at 13. And there could be something magical about 14, but I thought it more likely had to do with supplemental region pairing. As you can see, that 14 nucleotides is likely the shortest this target could be while still maintaining stable supplemental pairing. To further investigate the role of supplemental pairing, I carried out in vitro kinase assays using a series of length-matched targets with varying amounts of complementarity between the guide and the target. On top, I have the natural target I showed you before with the seed and supplemental pairing, followed by the seed-only, extended complementarity, and fully complementarity. When AGO2 is capable of slicing fully complementary targets, so I've included a catalytically inactivated version of Argonaute, shown in red, so that slicing doesn't interfere with the results. As you can see, only in the presence of this natural target with the seed and supplemental pairing do I see phosphorylation. telling me that supplemental pairing is indeed important for inducing the phosphorylation of Argonaute. But where exactly is this phosphorylation occurring? If we look back at the eukaryotic insertion, you'll see that there are 5 potential phosphorylation sites. And you can have different numbers phosphorylated, which leads to a lot of possible combinations. I wanted to see if I could narrow things down. I started by asking which can get phosphorylated. I took those 5 sites and I mutated 4 of them to alanine, so that they couldn't get phosphorylated. And I did this for each of the 5 combinations and tested their ability to be phosphorylated by CK1 alpha in vitro. When I did this, I saw that only 828 was able to be phosphorylated under these conditions. Next, I wanted to see whether 828 could prime for further phosphorylation because CK1 alpha is known to be capable of hierarchical phosphorylation, typically phosphorylating a serine or a threonine, 3 residues upstream of a previously phosphorylated site. To test this, I started with phosphomimetics, but these failed to induce further phosphorylation. However, phosphomimetics failed to recapitulate all the true features of phosphorylation, like size and charge. So I wanted to see if true phosphorylation of 828 could have a priming role. To test this, I developed what I call double-O mutants, which have 2 potential phosphorylation sites open for phosphorylation, 828 and 1 other site. I did this for each of the combinations. I would expect that if 828 could prime for the phosphorylation of the second site, I should see more phosphorylation when both sites were open compared to just 828 alone. And indeed, when I carried this out, I saw an increase in phosphorylation when 831 was open, leading me to propose that phosphorylation first occurs at 828 and primes with the phosphorylation of 831. The next question was whether 831 phosphorylation could prime for the phosphorylation of 834. To test this, I took that double-O mutant one step further, making it a triple O by opening back up 834 for the potential of phosphorylation by changing it back to its natural serine. When I did this, I saw a further increase in phosphorylation. This leads me to propose that 828 gets phosphorylated first, which primes the hierarchical phosphorylation of 831 and 834. So this tells me where on Argonaute the phosphorylation is occurring, but what effects is it having on Argonaute's activity? Specifically, I wanted to look at the effective phosphorylation on Argonaute's interaction with target messenger RNA. To do this using filter binding based target binding assays. I started with phosphomimetics. In the red, you'll see the 5XE phosphomimetic, mimicking a fully phosphorylated state. And in blue, I have the 5XA, which can't get phosphorylated. As you can see by the increase in the KD, the phosphomimetic has a lower target affinity. To see if this was just a function of charge alone, I tested a series of phosphomimics with increasing charge. As you can see, the effect is not purely additive, which suggests that certain residues or combinations of residues might have more of an effect, and I'm going to follow up with the phosphorylation sites that I identified. So that was phosphomimetics, but what about true phosphorylation? To test this, I took wild-type endogenously phosphorylated Argonaute, and I treated it with lambda protein phosphatase, a generic phosphatase that I found to be efficient at removing the phosphorylation from the eukaryotic insertion. When I did this, I saw a decrease in the KD, indicating that there's an increased affinity when you remove phosphorylation, implicating the phosphorylation having the negative effect. I don't have time to show you here, but when I treat the 5XA with the phosphatase, I don't see any effect. So this tells me that it's phosphorylation of the eukaryotic insertion specifically that's having the effect. I wanted to see if I could do a reverse. Instead of just removing the phosphorylation that was already there, could I add phosphorylation and see the decrease in affinity? This was more of a challenge because, as I showed you before, at least in my system, I need to have Argonaute already target bound before I can get it phosphorylated. So instead of studying target binding, I switched to a target exchange assay. I took Argonaute that was loaded with cold, unlabeled RNA, phosphorylated it with CK1 alpha, and then tested its ability to exchange that unlabeled target for a radio-labeled target. When I did this, I saw that treatment with the phosphatase increased the apparent KD, so it decreased the target binding affinity, and the opposite effect with the phosphatase. So this shows that it's phosphorylation through CK1 alpha mediated phosphorylation of Argonaute decreases its binding affinity. And this leads me to propose the following working model. Argonaute first binds to a guide microRNA, which it uses to take it to the corresponding target messenger RNA. Upon binding, once it's done, CK1 alpha comes and phosphorylates 828, followed by 831 and 834. Then, potentially because the negative charge of the target messenger RNA clashes with the negative charge of the newly added phosphates, the target gets released. This allows for regulation at the individual target level. But additionally, now that Argonaute is phosphorylated and has reduced target binding affinity for further targets, you get a global reduction in microRNA-mediated repression until PP6 phosphatase comes and resets the system, allowing Argonaute to bind normally again, until CK1 alpha comes back. And that leads to an outstanding question. What is it about binding to the target that makes Argonaute a good substrate for CK1 alpha? There are a couple of non-mutually exclusive theories I'm working with. One is greater accessibility. Argonaute is known to undergo conformational change upon binding to a target. As you can see in the structure of Argonaute bound to just a guide versus bound to a guide with seed and supplemental pairing, the PAS domain of Argonaute moves away from the region of the eukaryotic insertion, which is unresolved in the structures, so I'm showing it as a red dashed line. This could allow easier access for the kinase. Additionally, the negatively charged target backbone is in the region, which could serve as a recruitment mechanism for CK1 alpha, which is known to be acidophilic. I'm following up on both of these theories. With that, I'd like to say thanks to the Joshua Tor Lab, with special thanks to Lemur Joshua Tor and Alado Kayam. I'd also like to thank my school, Cold Spring Harbor Laboratories WSBS, as well as an NSF GRFP and funding from the Howard Hughes Medical Institute. With that, I'd like to take any questions.
Thank you for your talk, Brianna. That was really interesting. We do have one question in the chat. The Bashri is asking if you would please elaborate on the in vitro kinase assay.
Yeah, so basically I just take Argonaute that I've dephosphorylated and I mix it with CK1 alpha, ATP, and all the cofactors it needs. And then for these were endpoint assays. So I let the reaction go for an hour and then I stop the reaction, run them on an SDS page gel, and then use phosphor screen to measure the phosphorylation and quantify it that way.
Okay, I have another question from Thomas. He wants to know if there are any cysteines or methionine within the vicinity.
I don't know off the top of my head.
I have a few questions from William. Do you have a theory as to why the system exists? Is it perhaps to decrease affinity for binding to a target? And in general, how is CK1 alpha regulated?
Okay, so CK1 alpha is a ubiquitously expressed kinase, and it's one of a series, a family of CK kinases, and so this one is ubiquitously expressed, but I think that the target binding, because I think that the fact that you have to have the target binding in order to see phosphorylation serves as a sort of regulation mechanism so that CK1 doesn't just constitutively phosphorylate Argonaute. As to why this system would exist, I would imagine that it would allow Argonaute to access more targets if it is or has already been engaged with the target for a while. This would allow for an opening up. And if you could somehow have CK1 alpha like recruited to certain areas, you could imagine there being more of a multiple targets impacted at the same time in the same region, but not other places in the cell. So I think that's an interesting thing to think about.
Okay, I'll ask one more question. Are there other phosphatases such as PP1 or PP2A able to dephosphorylate?
I do not know. I know that, so the PP6 was identified in the CRISPR screen in that Golden et al. Paper as being the phosphatase, but I don't know if there are additional phosphatases that could play a role.
Okay, great. Thank you, Bri, for your wonderful talk.
Thank you.
Okay, our next speaker will be Dr. Subra Mandal from UT Arlington. I invite Subra to share his screen and begin his talk.
Can you see my screen?
Yes, you can see your screen. You'll just need to start the presentation and then you're good to go.
Is it looking okay right now?
Looks good.
Okay. So, Emily, for introducing, and also the ASBMB for giving me the opportunity to present. I'm Subra Mandal from Chemistry and Biochemistry University at Arlington. Today I will talk about a little bit of a recent work coding RNA, how it regulates, how it is associated with inflammation and glucose metabolism in macrophage. So before I go into my talk, I like to acknowledge, because at the end we don't have time for acknowledging the person who really did the work. There are two fantastic graduate students, Ira Obeid and Obishankar Chini. Primarily the work is done by Monira Obeid. She finished her PhD and now the project is taken over by Obishankar Chini. And there are many other people involved, but these are the two major graduate students who did all the work. So I think for this particular audience, I don't really need time to introduce non-coding RNA, but what I want to say that non-coding RNAs are a class of transcripts coded by the genome, but they are not mostly translated into protein. So even if these class of RNA, which are not converted into protein or translated into protein, they appear to have a critical function, and they are abundant in cells. They have tissue-specific expression, and that makes them attractive choice for developing potential biomarker or target for variety of disease. So in my lab, we work on gene expression, modification, and so on. So in the initial stage, many years ago, a graduate student who actually started looking for non-coding RNA, which are involved in breast cancer and regulated by hormones. And at that time, a non-coding RNA, hot air, was far expressed in breast cancer. And hot air is one of the, there are many non-coding RNA, and hot air is one of the long non-coding RNA, which is well characterized, and I think one of the most well-known non-coding RNA so far characterized. And because hot air is overexpressed in breast cancer, we thought we look at how it is regulated, what is its function. A little bit about hot air is that it's an antisense transcript, and it also interacts with two major class of chromatin modifying enzyme. One is called PRC2, which contains a histone methyltransferase EGS2, another a histone demethylase complex, which contains the demethylase called LST1. So histone H3 lysine 27 methylation by EGS2 and H3 K4 demethylation by LST1, both are linked to repression. The function of hot air is to bring this repressor machinery to the target gene or chromatin area, and does the demethylation and eventually suppress the gene expression. So classically, hot air is known to be, well, a repressor, a repressing non-coding RNA. And that's how we studied its function in variety of systems, and also because it is, in my lab, we show that hot air is required for breast cancer cell viability. It is also, its expression in breast cancer cell is also controlled by estradiol, and its expression is affected epigenetically by many estrogenic endocrine disrupting chemical like BPA, diethylstilbestrol, both in vitro, in cell culture, as well as an animal model. Now, today we will talk a little bit newer function of hot air. Although hot air is classically known to be involved in gene repression by chromatin modification, there appear to be a much more dynamic function. In this particular talk, I will talk about function of hot air in immune response and inflammation. It is very well known that during the infection or inflammation, there's a lot of change takes place, especially the upon bacterial or viral infection. There's a dramatic change in gene expression, especially the cytokine production. That includes interleukins, TNF, and so on. And the level of induction of these cytokines or chemokines are several thousand-fold. So interleukin 6 in response to infection is about 3,000 elevation. So because there is a dramatic change in gene expression, I thought hot air, being a classical repressor, it may be holding the genes or cytokine expression before infection at the repressed level, and upon infection, the repression is relieved so that there is induction in gene expression. With that anticipation, we started looking at the function of hot air. So we took some, as Monira did these experiments, she took some macrophage treated with lipopolysaccharide, which is the bacterial membrane mimic, and they bind to the receptor present in the macrophage, the immune cells, and once bind, there is a series of signaling cascade, and that leads to secretion of these cytokines and chemokines. So Munir took this macrophage treated with lipopolysaccharide, analyzed the cytokine expression, chemokine expression, and basically inflammatory response along with hot air expression. And you can see that when macrophage are treated with lipopolysaccharide, LPS, interleukin expression level is elevated tenfold, about 3,000-fold. In this oxide synthase, which is also a pro-inflammatory gene, it's increased. Along with, we see that hot air expression is increased. We are kind of anticipating that hot air will go down because it's a repressor, but instead it was increased similar to interleukins and nitric oxide synthase. So obvious question is, does hot air has a regulatory role in, or is it required for these cytokine expression? So next obvious experiment we did is knock down hot air in macrophage using siRNA and analyzed its impact on lipopolysaccharide-induced cytokine expression. Here is the real-time PCR data. Hot air expression is decreased, level is decreased upon siRNA treatment, and once the hot air level is decreased or knocked down, the LPS-induced interleukin 6 or nitric oxide synthase expression also went down. This suggests that hot air, instead of repression of these interleukins, rather it is helping their expression. It's a positive regulator in this cytokine expression. So this is very well signaling cascade involved in activation is very well known, and there's a transcription factor kappa B, NF kappa B, or NF kappa beta, which activation controls the cytokine expression. So there's receptors, they're recognized by lipopolysaccharide or infection, and that induces signaling cascade, which ultimately activates NF kappa B. And NF kappa B, once activated, translocates into the nucleus and binds to the cytokines or interleukins promoter, leading to the activation of the cytokine gene expression. So NF kappa B activation is a key or central to this inflammation or immune response induce cytokine expression. And our study shows that what here is required for this cytokine activation. So that means the next question is hot air, which is induced by lipopolysaccharide, does it control the NF-kappa B activation? We have done a series of biochemical experiments. I think here is a crucial data where we show that hot air is actually required for NF-kappa B activation. So this is a Western blot which shows that level of NF-kappa B in control and phospho-NF-kappa B in the control untreated cells and phospho-NF-kappa B, which is P65 in the LPS treated cell. You see the level of phospho-NF-kappa B is increased by LPS, that NF-kappa B is activated, but once we knock down hot air, the level of NF-kappa B activation goes down. So what it is telling that hot air in this case is controlling actually the NF-kappa B activation, and likely there's little bit more than likely by the degradation of inhibitor of NF-kappa B, which is called I-kappa B. I-kappa B gets degraded, and hot air somewhere is controlling the NF-kappa B activation via degradation of I-kappa B alpha. So I skipped this. So this is kind of the summary of this particular part, that NF-kappa B is a transcription factor. It remains normally complex I-kappa B alpha. When it is complexed with I-kappa B alpha, it is inactive. But upon infection or inflammatory response, this I-kappa B gets phosphorylated by its kinase, and this phosphorylated I-kappa B gets degraded by some ubiquitin ligases, polyubiquitination and degradation. And once I-kappa B alpha is degraded, NF-kappa B is released. NF-kappa B also gets phosphorylated and it translocates to the nucleus, binds to the target gene, and lead to the target gene activation. And target genes include interleukins, nitric oxide synthase, and cytokines. So basically, I-kappa B degradation controlling the NF-kappa B activation, and we are is somehow regulating the I-kappa B. At this point, we don't know whether it is regulating the phosphorylation or ignition. So that particular aspect is still being investigated. But at this stage, I want to introduce a little bit more on what goes on inflammation. Inflammation or immune response is a very important event, and it's metabolically also expensive because you need energy. And malfunction in energy metabolism has all kinds of disorders like obesity, diabetes, and neurological disorders. You can name almost every kind of these metabolic diseases which are linked to inflammation and signaling. So during this inflammation or immune response, there's a dramatic change happens in the metabolic metabolic programming, and especially in bacterial infection or innate immunity systems. Upon infection, the glycolysis pathway gets activated, which becomes the primary source of energy, and it's actually slows down the oxidative phosphorylation, beta oxidation, but takes over the glycolysis, which supplies the ATP. And there is other signaling factor which are critical mTOR or phosphofructokinase activation. Those activation is linked by this. So in our study, as hot air appeared to be a critical player in inflammation and cytokine activation, we immediately asked, does hot air may have any role in controlling these energy metabolism, especially in glycolysis? So we again took macrophage treated with LPS, and we looked at the expression of different glucose transporter, because glucose transporters are critical in importing glucose from outside to the cell, outside to the inside of the cell, so that the cells can do the glycolysis. So there are many glucose transporters. Those are called GLUTs, GLUT1, GLUT3, GLUT2, GLUT3, and so on. And we analyzed the several GLUT expression in macrophage in the absence and presence of lipopolysaccharide. And as you see in macrophage, the GLUT1, primarily the GLUT1 expression is elevated upon lipopolysaccharide treatment. So this, and also it is known that macrophage is primarily associated with GLUT1, and this is a Western blot which shows that GLUT1 level is also goes high, and that suggests that maybe hot air may be involved, because hot air is also upregulated by LPS. Hot air may be involved in regulation of GLUT1 expression as a function of inflammation. So we knock down hot air again in macrophage. We knock down hot air using siRNA and analyze the GLUT1 expression as dependent on LPS. So this is the hot air knockdown level, and this is the GLUT1 expression in the control LPS treated cell and under hot air knockdown condition and LPS. So this shows that upon hot air knockdown, the LPS dependent GLUT1 expression is also going down. And this is a Western blot which also shows that in the control GLUT1 expression is low. Upon LPS treatment, GLUT1 level goes high, and upon hot air knockdown, GLUT1 level goes down. This suggests that hot air actually regulates the glucose transporter GLUT1 expression in macrophage as a function of LPS stimulation or inflammation. So because the GLUT1 level went high and hot air regulates GLUT1, so
Subra, would you be able to wrap up in 1 minute?
Yes.
Keep on time. Thank you.
Last one.
Okay.
So because the GLUT1 level went high and hot air regulates GLUT1, we measure the glucose uptake, because that's the job of the GLUT1. So glucose uptake level in the control cell, LPS treated cell, glucose uptake is increased, and as we knock down hot air, the glucose uptake level, efficiency of the macrophage went down. That means hot air is actually controlling the GLUT1 expression as well as glucose uptake and eventually the glycolytic pathway. So this is the summary slide which shows that LPS induces NF-kappa activation with the coordination of hot air, and that controls the GLUT1 expression. GLUT1 is a glucose transporter, so GLUT1 elevation allows the glucose to come in, so the rate of glycolysis is increased. So with this, I think this particular talk is kind of concluded. At this point, my lab is moving, actually discovering a lot of new non-coding RNA which are in inflammation using RNA sequencing, and I don't think I have much time to talk. So maybe in the next ASBME meeting. So that critical player immune response, inflammation, and energy metabolism, in the process of discovering a lot of novel non-coding and linked immune response inflammation both in mouse and human system. With that, I would like to thank you for listening to me. Any question? I'll be happy to answer any question.
Thank you, Subra, for that wonderful talk. There are a number of questions. So someone is asking a question I'm also interested in is there a direct interaction between hot air and any of the NF-kappa B proteins, like a direct physical interaction?
That's what exactly we are looking at. We are doing hot air affinity purification and trying to especially look for ubiquitin ligases. And there are studies in neurological neuronal cell that hot air actually directly physically interacts with some of the ubiquitin ligases which control some of the neurological function. But it's not known immune response, inflammation.
Okay, and one more question. Is hot air only involved in the NF-kappa B pathway, or are there other immune pathways like the JAK-STAT pathway involved as well?
It is possible. It may be possible in other pathways. We have not looked at it.
Okay, thank you, Subra, for your talk.
Thank you.
I'd like to now reintroduce our chair, and our next speaker is Emily McFadden, who is a graduate student in Amanda Hargrove's lab at Duke, who's going to tell you about analysis of long non-coding RNA structure-function relationship in prostate cancer.
Subra, I just need you to unshare your screen. Okay. Thank you all for being here today. And thank you to the ASBMB committee for giving me the space to speak. I'm excited to present my work on long-noncoding RNA and its role in prostate cancer. To give you some background on prostate cancer, it is the second leading cause of death in American men and accounts for 10% of men's death for ages 65 plus and 20% for 85 plus. Overall, 1 in 9 American men will develop invasive prostate cancer in their lifetimes. Unfortunately, like most cancers, this disproportionately affects marginalized groups, where Black men are 60% more likely to develop prostate cancer than white men. I also want to point out that the poor survival rate for prostate cancer is largely attributed to distant or metastatic prostate cancer, where the survival rate drops from nearly 99% survival to as low as 20% survival when the cancer metastasizes. Although there are many factors that contribute to the initiation and progression of prostate cancer, today I will focus on the role of long-noncoding RNA in metastatic prostate cancer. The first link RNA I will discuss is MALAT1, shown here on the left. MALAT1 is overexpressed in prostate cancer tumors as compared to normal prostate tissue, and these higher levels of MALAT1 expression correlate with poor patient longevity as plotted on this Kaplan-Meier curve. As I will explain in more detail soon, MALAT1's longevity in the cell is in part attributed to a triple helix at the 3' end of the transcript that acts as a molecular knot to hinder exonuclease degradation. The other link RNA I will discuss today is SHLAP1, shown here on the right. Similar to MALAT1, SHLAP1 is overexpressed in prostate cancer tissues compared to normal tissues, and higher levels of SHLAP1 again correlate with poor patient longevity. There is some information regarding the interaction between SHLAP1 and chromatin remodeling complex SWSNF that I will discuss later. In general, long-noncoding RNA or link RNA structure-function relationship in cancer is poorly understood, and although we know both that MALAT1 and SHLAP1 are overexpressed in metastatic prostate cancer, their direct role in the disease pathway is not known. The question I'm trying to answer and will present data on today is what are the roles that MALAT1 and SHLAP1 structure play in metastatic prostate cancer? I will start my talk with MALAT1. So this link RNA is over 6,000 nucleotides in length and is known to associate with nuclear speckles and be involved in post-transcriptional processing. The knockdown of MALAT1 reduces migration of prostate cancer cells and increases their apoptosis. This metastatic phenotype was localized to the last 1,500 nucleotides of MALAT1, and further investigation of the 3' end through both bioinformatics and crystallography determined a triple helix structure. Interestingly, transcript accumulation can be reversed through either deletion of this triple helix gene structure or partially reversed through a single point mutation that would reduce the stability of the triple helix. Based on this information, dysregulation of triple helix stability could promote transcript degradation and reverse our cancer phenotype. To build on this hypothesis, we are investigating the role of MALAT1 3' end triple helix dynamics in relation to transcript stability, as well as with small molecule binding. I will only be discussing the work today related to dynamics, but I've included some information about our seminal paper that identified the suite of small molecules that bind to the MALAT1 triple helix with nanomolar binding affinity. While the crystal structure I showed you earlier indicates that the 3' end of MALAT1 exists as a triple helix, the work of Baird's lab used FRET-labeled two-body system MALAT1 triple helix to propose that there are multiple conformations that exist in solution. Notably, this work identified that there's a pre-organization of the UU base pairs as a duplex prior to association of this A-rich tail that forms the triplex. This work is supported by our own native gels, where we use both a two-body system as well as the triplex wild-type system at different salt conditions, 0 and 1 millimolar magnesium, to observe multiple conformations of the RNA, as seen by the distinct bands. However, both of these work lack high-resolution solution state structural information of the native triple helix to identify what these distinct states are. We thus collaborated with the Al-Hashimi lab at Duke University to use SOFAST amino 1D proton NMR and see if we could identify multiple dynamic states of the MALAT1 triple helix using the crystallography construct, as the MALAT1 wild-type construct is too large for NMR as a first pass. Within this first experiment, we were looking for a change in the signal at the amino region between 10 and 12 ppm. In the duplex state, the UU base pairs will persist. As the triplet forms, a UAU, wobble, and Watson-Crick base pairs will form, and the amino signal will disappear. We used two temperatures, 25 degrees Celsius and 11 degrees Celsius, to attempt to observe these conformations. Unfortunately, even the truncated crystallography construct is too large and dynamic to identify distinct states at any temperature, but these data support the existence of multiple states for the uridines participating in the triplet bonds. In summary, Baird's FRET work supports the existence of multiple MALAT1 conformation, and our own work supports this as well. However, we require improved resolution to identify what these distinct structures are. So further work will build on this progress and use site-specific labeling to improve resolution, identify the multiple conformations of the MALAT1 triple helix, and also provide a tool for my coworkers to screen other small molecules that may bind to the MALAT1 triple helix. In the second half of my talk, I will discuss the long-noncoding RNA SHLAP1. So this work was done in collaboration with Chennaiyan at University of Michigan, and they were the first to identify SHLAP1's relevance to prostate cancer. This work, among others, showed that prostate cancer cells with overexpressed SHLAP1 injected into mice to form different metastases, as seen in this orange box over here. These cells formed fewer tumors in mice when a stably expressed shRNA targeting and promoting the degradation of SHLAP1 was transduced into the cells, seen in both the green and blue boxes. The Chennaiyan lab completed deletion studies of SHLAP1 to investigate if a particular region of the link RNA is important for this proliferative phenotype. Interestingly, only a single 250 nucleotide deletion, deletion 5 or D5, was found to inhibit this cancerous phenotype. Additionally, this deletion mutant was unable to pull down the SNF5, a subunit of the SWI/SNF complex, which is a chromatin remodeling complex that normally associates with SHLAP1, as seen over here. From this work, we and others hypothesized that SHLAP1 overexpression interferes with the normal function of the SWI/SNF complex in prostate cancer metastasis, and that the deletion 5 mutant inhibits the association of SHLAP1 with the SWI/SNF complex. Knowing that this 250 nucleotide region is important for protein association and proliferation, how does this deletion mutant change the structure of SHLAP1, and is this fragment sufficient for protein association or proliferation? To begin, I first completed chemical probing of SHLAP1 wild type and deletion 5 using shape reagent. Shown here is a secondary structure model informed by the chemical probing for just the wild type construct. Because these experiments were completed in vitro and we wanted to preserve the most native structure possible, we were curious how the purification of the RNA might affect the structure. Here I'm comparing the natively purified RNA that was probed versus RNA that was denatured, annealed, and then probed. So Delta Shape is a program that compares the chemical probing reactivities of two RNA structures and identifies statistically significant differences between the two. Each peak on this graph corresponds with a single nucleotide whose reactivity is statistically significantly different And the directionality, either positive or negative, refers to which sample was more reactive, either the annealed or the native. Here I'm comparing the two wild type samples. It is interesting to note that regardless of the sample preparation, the D5 region within the wild type transcript over here shows no difference. And overall, less than 8% of the nucleotides display statistically significant differences between the two sample preparations. The large region in the wild type that does show differences is noted with the star over here, did not drastically alter the minimum free energy structures that were predicted. And when I analyzed them using a SymTree computational program, I saw very few differences between the two. Overall, the SHLAP1 secondary structure is well maintained regardless of sample handling. So we chose to use the native preparation of both the wild type and D5 for our future analyses. Here I'm showing you the rainbow arc plots for the wild type and D5 constructs. The alternating fragments correspond to the different deletion mutants created by the Chennaiyan lab. We can see in the first 1,000 or so nucleotides that there is a large secondary structure arc that exhibits a lower probability in the deletion mutant in yellow, as it does to the wild type in blue on the bottom. In red over here is the region that is deleted in the D5 mutant, seen on top. Interestingly, interactions that normally occur between the D5 and D3 region persist in the D5 mutant, but have changed between rather D3 and D5 to D3 and D6. This could explain why the other deletion mutants do not display phenotypic changes. With this in mind, we chose to zoom in on the deletion 5 region and investigate it more. So this is just a zoom in version of the slide I was just showing you. And we identified in this D5 region two highly predicted secondary structures that we termed ARM-A and ARM-B. These two highly structured regions are found in the majority within the D5 region, but expand into D4 and D6. However, I want to reiterate that neither D4 nor D6 reduce the proliferative phenotype that I showed you earlier. So here I'm just mapping those two structures onto the secondary structure wild type version. And we can see that, again, these two structures are highly zipped up, well base paired regions. Along with my coworker, James Felice, we have transcribed and purified these regions, ARM-A, ARM-B, and both the full-length ARM-A plus ARM-B for independent chemical probing experiments. We are interested to see if these well-predicted structures are able to fold on their own and may constitute an independent folding domain that is critical for the SHLAP1 cancer phenotype. Additionally, James and I are expanding on the shape chemical probing and incorporating DMS probing to further enrich our models. In summary, we have determined the first secondary structure model of SHLAP1 wild type and D5 and identified a putative independent folding domain that may be related to the cancer phenotype through interactions with the SWE/SNF chromatin remodeling complex. We will build on this work by improving our model through the addition of more chemical probes such as DMS, characterization of the independent folding domain, and seeing if this IFD is sufficient for SNF5 protein pull down or cell proliferation. From this work, I hope you appreciate the relationship between long-noncoding RNA structure, function, and prostate cancer, where I specifically highlighted MALAT1 and SHLAP1. I want to thank you all for your time and attention, and I would be happy to take any questions.
Thank you, Emily. That was a great talk. And I really liked some of those graphics. It was really cool. So we have a few questions. Does MALAT1 have different RNP proteins associated with it in the different conformations?
That is a great question. We don't know. We do know whether proteins are associated in the cell or not. There's a couple of the methyltransferase, the metal family proteins that are associated with MALAT1, but we do not know if those are conformation dependent at this time. That's a great question.
Okay, another question. Can D-shape predict non-coding RNA mRNA interactions?
So delta shape is meant to compare the secondary structure, the chemical probing reactivities of two different RNAs with each other to see which one has basically the differences in probing single-strandedness or base pairedness between two RNA structures. It's not meant to look at the association of RNAs with each other in the cell. So I'm not sure which program you would use to look at mRNA link RNA associations in the cell directly.
So Thomas is asking if any specific species or mutant. I'm not exactly sure. This was in the second half, so I think he was asking with your SHLAP one if it was which species you used it from.
So to date, SHLAP1 has only been identified in humans. So we're using the human version of SHLAP1 isoform 1 for our studies.
Is the secondary structure formation in link RNAs context dependent?
I'm not quite sure what that question is asking. So I will speak broadly about it. So it's proposed by a number of different labs, the Sanbamatsu lab and the PIO lab, that structures within link RNA are formed in these independent phylodomains. So really highly structured modular regions of link RNA. And these independent phylodomains are often related to specific functions, namely protein binding. So of note, the PIO lab, going off of the hot air talk we just saw, identified an independent phylodomain important for PRC2 interaction, which is then related to its function, as we just learned about. I hope that helps answer the question.
How stable are the alternative conformations? I'm assuming he's referring to the mallet one.
Mm-hmm. So based on the NMR, we don't see distinct states with our NMR technique that we used. So there's probably a lot of flux between the two or more different states that exist in solution. If we're talking about in terms of population frequency, for multiple RNAs, these can range from as much as 50% to as low as 0.1% of the population. But regardless of population frequency, they still have important functions in the cell.
I have a question. Have you tested at all changes with pH and folding?
Changes of pH and folding. We have not explored pH. The chemical probing, at least for the chemical probing experiments, the pH is tightly regulated, and that's in part due to the sensitivity of the probing reagents to being hydrolyzed in solution. For the NMR experiments, we have not explored the role of pH, but we are interested to look at mallet-1 dynamics at both lower and higher pH to see if that could affect the triplet stability and allow us to see different states more easily. Yeah.
Is there known genetic variation or SNPs in cancer that can change the structure?
Yes. I know this specifically for mallet-1. There are a number of SNPs that occur in the triplet region of this 6,000 nucleotide transcript that do alter the stability of the triplet, and that's found in a number of other works that I didn't show you today. But just as that U to C mutation reduced the stability, there are a number of other mutations in that same region, both in the A tail or the UU base pairs that are naturally occurring.
For the conformational changes, does it require some other cofactors such as an RNA chaperone?
That's also a great question, but we don't know that to date. That's a longstanding question is, does the RNA sample multiple conformations, which then enables it to bind to protein in a specific conformation? Or does the RNA bind to a protein and then change conformation? So that's something we don't know right now, but that's something we're very interested in.
How sensitive are the conformations to RNA exosome processing?
Can you repeat that question?
How sensitive are these conformations to RNA exosome processing?
I don't feel like I can answer that question. I'm sorry, I don't know.
Okay, I think that is all the questions. So thank you.